Here are some notes on enzyme structure and function. Chemical reaction is a process that changes or transforms one set of chemicals into another. The elements or compounds that enter into a chemical reaction are called reactants. They appear on the left side of the arrow. The elements or compounds that are produced by a chemical reaction, those are called the products, and they appear on the right side of the yield arrow. So looking at this, you would read it, H2O2 yields H2O plus O2, where the H2O2 is the reactant, the arrow means yields, and the products are H2O2 and O2. H2O2 is peroxide, hydrogen peroxide, H2O is water, and O2 is oxygen. Chemical reactions always involve changes in the chemical bonds that join atoms and compounds. So energy is released or absorbed whenever chemical bonds are formed or are broken. Because chemical reactions involve breaking and forming bonds, they involve changes in energy. Some chemical reactions that make life possible occur too slowly to make them useful for organisms. Catalysts are chemicals that speed up the rate of reaction, and enzymes are proteins that act as catalysts. So enzymes speed up chemical reactions that take place in our cells. Enzymes provide a site where reactants can be brought together to react. And the reactants of the enzyme catalyzed reactions are called substrates. So the reactant of the enzyme catalyzed reaction is called a substrate. And enzymes are very specific, usually catalyzing one reaction only. So enzymes are based on design, and so they are very specific to their the site or their substrate that they will bind to. Enzymes function on what we call a lock and key model. Like a lock, it's specific to a key, to the key that it is opened with. The enzyme is specific to the substrate it reacts with. And so if we look here, if this is only this key will fit into this lock based on the shape, only this substrate will fit into this enzyme. A substrate is what will find the enzyme that breaks it down. So the substrate will find the enzyme. And so looking here, the enzyme kind of looks like a puzzle. It has some carvings in it. These substrates will find this enzyme, and they will form an enzyme substrate complex. Okay, so they will find the active site within the enzyme. They will then bond, forming what we call an enzyme substrate complex. So this is an intermediate product. The substrate is changed into a product. So these substrates, we start with two of them, is changed into one product. And notice that the enzyme is unchanged. So the enzyme is unchanged and therefore it's reusable. So we're looking at the function of the enzyme. Let's take water and carbon dioxide, for example. Water and carbon dioxide, those substrates will bind to the enzyme here at the active site, which is this opening. They will form this enzyme substrate complex. The substrates are converted into products. So old bonds are broken, new bonds are formed. The products then get released in the form of carbonic acid as our new molecule. And notice that the enzyme, which is carbonic anhydrase, anything that ends in ASE is an enzyme in biology class, so anhydrase, carbonic anhydrase. This enzyme has the same active site and after that product is formed and then released. So it remains unchanged and can be used again to then catalyze more water carbon dioxide reactions in order to produce that carbonic acid. The proper functioning of enzymes depends on the proper shape of the enzyme. So when an enzyme loses its shape, we call that denaturing. So it's said to be denatured and the enzyme stops working. Enzymes require a specific pH, temperature, and concentration to work at their optimal or best level. So these three variables, pH, temperature, and concentration, can affect the efficacy or how efficiently the enzymes do their job. So if we look at enzyme function, okay, and we've said before that proteins are unique because they are three-dimensional in character, and active proteins properly fold in this manner. When heated, however, and enzymes are an example of a specialized protein, when heated, they become inactivated if they are heated to a particular temperature. That will cause them to denature, and they'll unravel, and then their unique 3D structure will be changed, and we say that this inactive protein has denatured, so it has due to heat, due to a change or a very high temperature. So that's the end of our enzyme structure and function notes, and we will continue learning about enzyme structure and function and the variables that affect it in the lab that will test the pH and the temperature and concentration of the enzyme on its ability to function.